Hello, everybody. I am Dr. Lukena Karkhanis. I am an allergist, an immunologist. I practice in San Antonio, Texas, and I'm very happy to have the opportunity to address you and give you a little bit of information about the current COVID-19 vaccines. So, like we say in immunology, you can memorize everything and still understand nothing. Immunology is very complicated, right? We are going to try and understand a little bit about the immunology of COVID-19 vaccines in a concise presentation. Let's start with some information about the SARS-CoV-2 virus. So it's serious acute respiratory syndrome coronavirus 2, SARS-CoV-2. This is the causative agent of COVID-19. It is from a family called Coronaviridae, and they are a Baltimore class 4 positive sense single-stranded RNA viruses. They don't have any DNA. Their RNA is about 30,000 bases in length, and the entire virion is about 50 to 200 nanometers in diameter. So it's very small particles we're talking about here. They have 4 main structural proteins. The spike protein, which you can see as the big red spikes in this pictographical depiction, the envelope protein, the membrane protein, which are the smaller yellow and orange spikes, and then inside you have the nucleocapsid proteins. And they utilize various receptors in our body, the ACE2 receptors and bassigin. How about what happens when this virus enters our body? So how is DNA converted to mRNA and to protein in our body? This review is important because we are talking about DNA and RNA vaccines here. As you know, our genetic information is stored in a double helix strand of DNA. When it's time for a particular part of the DNA, the blueprint, if you will, to be converted to actual protein, it unwinds and gets converted through transcription to a single-stranded RNA called the mRNA. The mRNA then leaves the nucleus, it comes outside, and it is taken up by a ribosome complex, another protein organelle in our cell, and translated into protein. So every 3 base pair of mRNA is translated into a particular peptide, and then the amino acid, and then the amino acids join to form a polypeptide chain, which then folds to form the actual protein. So this is how we are made, every part of us, our hair and our skin and our blood and our organs and our enzymes and hormones. This is how every part of us is made. DNA, which is the blueprint, gets converted to mRNA, which is kind of the standard operating procedure instructions, or SOP, and that gets converted into actual protein, which are the building blocks of the human body. How does SARS-CoV-2 infect a cell? So again, a quick pictographical depiction. On the left hand, you can see the SARS-CoV-2 virion approaching the cell. The spike proteins, the little projections shown here on its surface, they catch on to the receptors. One of the main ones is the angiotensin converting enzyme 2, or ACE2 receptors. That is what allows the virion to anchor onto the surface of our cell, and then the membrane of the viral particle fuses with our cell membrane, and the contents of the viral particle are able to enter our cell. All viruses essentially hijack our cellular machinery once they enter our cell. They don't have any protein making machinery of their own. They don't have ribosomes. What they do is they hijack our cell machinery, and so the RNA inside the cell is taken up by our ribosomes, and our ribosomes, instead of making our own proteins, start making viral proteins. They start making all the nucleocapsid proteins, spike proteins, membrane proteins, envelope proteins. And since we are talking about a live COVID-19 infection, the RNA replicates as well, and more pieces of RNA are formed. These are then assembled in our cellular machinery into new viral particles, which then get released from our cells. They go on to infect the neighboring cell. So essentially, this is how a virus spreads inside a host. It infects a cell, replicates inside that cell using that cell's machinery, and then goes on to infect neighboring cells. What are vaccines? So a vaccine is a biological preparation that provides active acquired immunity to a particular infectious disease. Typically, it contains an agent that resembles the disease-causing microorganism, and vaccines are often made from weakened or killed forms of that microorganism, or its toxin, or one of its surface proteins. This agent in the vaccine stimulates the body's immune system to recognize that agent as a threat, destroy it, and then to remember and further recognize and destroy any microorganism associated with that agent that it may encounter in the future. So this is essentially like a military training exercise for the soldiers of your body. They go out, they see a pretend enemy, they fight it, and they learn to remember what it looks like so they can fight it more effectively in the future if they encounter it again. Vaccination is the most effective method of preventing infectious diseases, and widespread immunity due to vaccination is largely responsible for the worldwide eradication of many diseases, like smallpox, restriction of many diseases such as polio, measles, and tetanus from much of the world. Licensed vaccines are currently available for 29 different human infections. We also have many other vaccines for animal infections. What are the basic types of vaccines? You can have a DNA or RNA vaccine, which we now have for COVID-19. Essentially, this vaccine uses DNA or RNA molecules to teach the immune system to target key viral proteins. You can have live attenuated vaccines, which introduce a weakened form of the virus into our body. Examples are the oral polio vaccine and the nasal flu vaccine. You can have an inactivated vaccine where the virus has been killed and the killed virus is being introduced into the body to train the body to fight it. Examples are Covaxin, the Indian COVID-19 vaccine, our injectable flu shot, our injectable polio vaccine. These are all inactivated vaccines. There is a type of vaccine called a subunit vaccine, where just a piece of the microorganism's surface is used to focus the immune system on that target. Examples are the hepatitis B vaccine. And then you can have a viral vector vaccine, for example, the AstraZeneca Covishield and the Johnson & Johnson Janssen, where you take a harmless virus, introduce your COVID-19 virus inside that harmless virus, and you use that harmless virus as a way of training the body to fight COVID-19. So let's first look at mRNA. How do mRNA vaccines work? For example, the Pfizer and Moderna. So on the left hand here, we have a pictographical depiction of an mRNA vaccine. Essentially, it has the RNA sequence of the spike protein of COVID-19 inside a lipid envelope. Why a lipid envelope? That's because spike protein mRNA, in general, all mRNA, all RNA is very, very fragile. Scientists who work with mRNA can tell you that it gets destroyed in seconds if it's not kept on ice and handled very, very carefully. The entire world, our bodies, our body surfaces have RNA destroying enzymes called RNases in abundance. And so to protect this RNA from being destroyed outside of our body, we had to put it in a lipid envelope. And that's why these mRNA vaccines have a lipid envelope. This, when introduced into the body, the lipid envelope fuses with our cell membrane. And that RNA, which contains the genetic sequence for the COVID-19 spike protein, gets released into the cell. It hijacks the cellular machinery. The ribosomes that were making the cell's proteins now see this little piece of RNA floating around, and they decide to make it into protein, because that's what they do. They see RNA, they make it into protein. So they start making spike protein. This spike protein then is released onto the surface and waits for the immune cells of the body to come recognize it as a threat, as a foreign invader, and to mount an immune response. And then on the lower part of the diagram, you can see some of that spike protein, the cell does recognize as foreign, and so it starts chewing it up in these cellular complexes called proteasomes. It gets chewed up into peptides, small tiny pieces. These peptides then get loaded onto very specific structures on our cell surface called the MHC receptors, which then further aid the immune cells in recognizing that this is a foreign threat. How do viral vector vaccines work? For example, the AstraZeneca Covishield vaccine and the Johnson & Johnson Janssen vaccine. So these vaccines have a harmless, non-live, non-replicative adenovirus which cannot infect human beings. So for example, a chimpanzee virus, inside that virus they have put the DNA for the COVID-19 spike protein. This harmless non-replicative virus then goes inside the body and it fuses with the cell membrane and it releases that spike protein DNA. It goes into the nucleus, because remember these viral vector vaccines are DNA. They go inside the nucleus and that DNA gets converted into mRNA for the spike protein. It then comes out of the nucleus, sees ribosomes, and the ribosomes start converting it into protein, which is the spike protein. And the spike protein then is either exposed on the surface or is chewed up by the proteasome and is mounted onto the MHC complexes. Now you have to keep in mind, though the DNA enters the nucleus, it cannot go anywhere close to our DNA. Our DNA is inside our nucleus, but it's very, very tightly bound and regulated and protected by proteins called histones and other complexes. So it's akin to being inside a safe. So just as you know, any random piece of paper that enters your house cannot make its way to your safe. Somebody that needs to know where the safe is, needs to know how to unlock the safe, needs to want to put that piece of paper into the safe, is required to actually put that paper into the safe. And that's the same thing with our DNA. So all the mounting concern about these vaccines integrating the viral DNA into our DNA are not founded on any kind of scientific basis, because keep in mind, it is very hard to change our DNA. Here we are trying our best not to change our DNA. There are other instances where we are trying to change our DNA, like gene therapy for genetic diseases, and we still haven't been successful. That is how hard it is. Here we are trying our best not to do it, and so it's not going to happen. This DNA has no enzymes required to break through the barriers around our DNA to go anywhere close to our DNA. And what do you think happens when we catch infections with viruses that have DNA? This is the exact same process that happens. These viruses are attacking us 24/7, 365. So it's not like this process started inside our cells now because we made a COVID-19 vaccine. Our cells are constantly seeing viral RNA and DNA and making viral proteins, and our immune system mounts a response and we don't fall sick. If this had changed our DNA to any significant extent, we would have all died in childhood, given how many viruses we fight on a daily basis. So there is no need to think that this DNA goes anywhere close to our DNA. How do inactivated vaccines work? For example, the Bharat Biotech Covaxin. So here what they did is they took the entire COVID-19 virion and they killed it with a chemical so that all the proteins and the RNA is inactivated. It's no longer live. And then that is injected into the body. The immune cells see it as debris, as a foreign invader, or something that's a danger, and they start taking it up and breaking it up because they need to break up the trash and they need to get rid of the trash in the body. And in that process, they break it up into the individual proteins and they expose them on the surface, and the immune cells get trained to fight all the proteins. So one of the advantages of the inactivated vaccines is that the immune system learns to fight against not just the spike protein, but all the proteins. So all these vaccines have their own pros and cons. So how is the immune response stimulated during any vaccination or infection? you have the antigen. In this case, we saw how the spike protein is made. That antigen is exposed on the surface of those muscle cells that have taken it up. The antigen then interacts with a very specific immune cell called the antigen presenting cell, and these are your macrophages and dendritic cells. These cells have a specialized way of taking up that information from that antigen and passing it on to our immune cells, the soldiers of our immune system, which are our lymphocytes. They are a type of our white blood cell, and there are two types. There are B cells and there are T cells. The B cells then go on to mount what's called a humoral immune response. They produce antibodies. So all this talk we hear about antibodies to COVID-19, it is our B lymphocytes that are making these antibodies, IgG, IgM, IgA. They go on to produce these antibodies which float around in the body, looking out for the next spike protein to attack it and inactivate it. Some of these B cells go on to form memory B cells that will be stored in the body, so that the next time the body sees this same spike COVID-19 protein, it doesn't have to start from scratch. It has these already trained soldiers ready to just start replicating and deploying and producing massive amounts of antibody. The other type of cellular response is the T cell mediated response. These T cells turn into what are called killer T cells and helper T cells, which then go on to destroy the cells that have been infected with the spike protein COVID-19. They also form memory cells and linger in the body for years, so that the next time they encounter a cell infected with the COVID-19 spike protein, they already know how to fight it and they can destroy it quickly. So these are the main types of immune responses we would see to essentially any vaccine or virus. And let's look a little bit more in detail to this last part of the immune response. So you can see that the viral antigens are being presented on the cells. They also go through a proteasome, get chewed up into small peptides, and in the muscle cells where you get the vaccine, they get loaded onto a protein complex called MHC1, major histocompatibility complex 1 protein. Every cell in our body that has a nucleus has a MHC1 protein on its surface. This is what we type when we do typing for transplant, because MHC, major histocompatibility complex, is kind of the identity of your cell. That MHC1 peptide complex is then recognized by a specific type of CD8 T cell, where this cell with this MHC1 with its peptide keeps floating around, and the CD8 cell has a T cell receptor, and when a matching receptor finds this MHC1 peptide complex, it binds it, it gets stimulated, and it starts replicating. The second thing that happens is when this spike protein is taken up by those antigen presenting cells, the dendritic cells, and taken to the lymph node, it goes through a lysosome complex and gets loaded onto MHC2 complexes, major histocompatibility 2 complexes, which are present only on these antigen presenting cells. And those then are recognized by CD4 T cells and stimulated. Some of the spike protein is directly exposed on the surface and recognized by B cells via their B cell receptor. How do they recognize this? You have to keep in mind T cells and B cells are formed in our body and are formed with millions of clones available. So all of us, many years ago, even before COVID-19 came into the human population, have had these clones that could recognize COVID-19 peptides just floating around, waiting for the day that they will find their match. They are formed by very specific genetic recombinations in the genes that encode these T cell and B cell receptors. And so essentially we are born with the ability to form T and B cell receptors that match millions of peptides, and they just float around till the day they find their matching peptide. So it's not like our body started forming these now after COVID-19 came into being. We have always had them. They've just never had an enemy to fight. Now they do. They recognize the spike protein peptides, and they realize that this is what they were made for. This was their calling in life. They start replicating, start converting into memory cells, and start mounting an immune response. Let's look at the COVID-19 vaccines in the United States. So in December 2020, the US FDA granted emergency use authorization to Pfizer-BioNTech for age 16 and above and Moderna for age 18 and above for their COVID-19 vaccines. These are both mRNA vaccines. They have no live virus. They are enclosed in a lipid nanoparticle. They are preservative-free, and millions of doses have been administered in the US to date. In February 2021, the US FDA granted emergency use authorization to Johnson & Johnson for their COVID-19 vaccine for age 18 and above. This is an adenovirus vector vaccine, no live virus in it, and it is again preservative-free. And several million doses of this vaccine have also now been administered in the US to date. What about the COVID-19 vaccines in India? In January 2021, India's DCGI approved AstraZeneca's Covishield and Bharat Biotech's Covaxin for use. Covishield is an adenovirus vector vaccine, like we discussed, replication-deficient chimpanzee common cold virus with the SARS-CoV-2 spike protein gene inside it. It has no live virus. Covaxin is an inactivated SARS-CoV-2 virus vaccine. So they took the virus and they doused it with a chemical called beta-propiolactone, which binds to the genes and inactivates replication but leaves the proteins intact. It has no live virus. It does have an aluminum-based adjuvant called alhydroxyquim-2. Why an adjuvant? An easy way to understand this is, if 4 mosquitoes enter your house, are you going to call the cops? No. But if 4 strangers with weapons enter your house, are you going to call the cops? Of course. So size matters in terms of danger signals and immune responses. So when we are trying to get the immune system to respond, we have to make the target big. We have to make it look dangerous. So we have to add adjuvants to make sure that our immune system doesn't ignore it as a tiny non-threat. Let's look at a quick vaccine comparison. Okay, so we have Pfizer, Moderna, Johnson & Johnson, Covishield, and Covaxin. I concentrated on these because these are the ones we've kind of talked about so far. Pfizer and Moderna are mRNA. J&J and Covishield are adenovirus vector, and Covaxin is inactivated virus. Pfizer had 43,000 participants in its trial, Moderna had 30,000, J&J 40,000, Covishield almost 24,000, Covaxin almost 26,000. So thousands of volunteers in these trials. So all of these vaccines have been very well studied. J&J was studied for single dose, but the other 4 all consist of 2 doses. Pfizer, at least 7 days after second dose, was 95% effective in preventing symptomatic lab-confirmed COVID-19 infection. Moderna, at least 14 days after second dose, was 94.1% effective. J&J, at least 14 days after their single dose, 66.9% effective in preventing moderate to severe COVID-19. They didn't really look at mild cases. So each of these trials varies a little bit, and it's hard to compare them head to head. Covishield, after 2 doses, vaccine efficacy was 70.4%, and after at least 1 standard dose, it was 64.1%. So AstraZeneca's trial data is a little more complex because they had different doses and they had different regimens. Some people got a low dose, some people got a standard dose, and some people got 1 dose, and some people got 2 doses. Covaxin, their phase 3 trial data has not yet been published, but in their press releases, they claim that at least 14 days after second dose, they were 80.6% effective in preventing symptomatic lab-confirmed COVID-19 infection. All the vaccines claim similar efficacy across age and sex and race and ethnicity and underlying medical conditions. In Pfizer, there were 8 COVID-19 cases in vaccine group and 162 in placebo group, so far fewer in the vaccine group. Moderna, 11 in the vaccine group and 185 in the placebo group. And they even looked at asymptomatic infections in a small group, and there were 14 in the vaccine group and 38 in the placebo group. Johnson & Johnson, 116 cases in the vaccine group, and they are looking only at moderate to severe, 348 in the placebo group. Covishield, 63 in the vaccine group, 150 in the placebo group. And Covaxin, interim analysis, 7 cases in the vaccine group, 36 in the placebo group. And they claim they will not release their data till they meet a certain minimum number of infections. What about severe infections? So Pfizer, there was 1 in the vaccine group and 3 in the placebo group. Moderna, 0 in the vaccine group, 30 in the placebo group. But if you read the footnotes in Moderna, there was actually 1 case that required hospitalization in the vaccine group. And if you go back to Pfizer, that 1 severe case in the vaccine group was deemed severe just based on 1 pulse ox reading, but otherwise behaved like a moderate case. So very low numbers overall. J&J, 2 people were hospitalized with COVID-19 in vaccine group, 29 in placebo group. Covishield claimed 0 severe cases in vaccine group, 2 in the placebo group, but this data is before the variants popped up. They have not put out their exact data after the variants came out. These numbers are probably different. Let's look at side effects. So for all the vaccines across the board, the most common solicited adverse effects were what we kind of see with any vaccine. Injection site pain, injection site reaction, fatigue, headache, muscle pain, chills, joint pain, low-grade fever, myalgia, tenderness, feeling feverish, malaise. Lymphadenopathy has been reported in the mRNA vaccines. And in fact, radiologists here in the US are recommending that people don't get routine mammogram screening right after getting their Pfizer or Moderna vaccines, because you often have incidental finding of axillary lymphadenopathy. There were 4 cases of Bell's palsy in the vaccine group in Pfizer, none in the placebo group. But the 4 cases in the vaccine group still don't represent a frequency above that expected in the general population. So the feeling is that this was a coincidence, but we will study this as the vaccine rolls out. There are no indications from VAERS data with millions of doses given out now that there is any change in this. In Moderna, there were 3 cases of Bell's palsy in the vaccine group and 1 in the placebo group. In J&J, 2 cases in the vaccine group and 2 cases in the placebo group. In Pfizer, there was 1 case of shoulder injury, and that's why it's important that the person giving the injection know where to give it. Interestingly, in the trials, there was no anaphylaxis reported, but there is some data coming out from VAERS of a small number of cases of true anaphylaxis to the vaccine. But that is not unexpected. We are never going to have a vaccine or a medication that has 0 side effects. No deaths due to any of these anaphylaxis reactions. In Moderna, interestingly, there are a few reports of facial swelling in participants who had had dermal filler injections. So dermal fillers are a cosmetic procedure that is done for wrinkles and for aging. And dermatologists will tell you that oftentimes even viral infections like flu can cause swelling at the site of previous dermal filler injection, and it seems to be due to inflammation at the interface between the filler and the body. And they did see, interestingly, 3 cases of swelling in the Moderna trial. They did see some intractable nausea, vomiting, and facial swelling, but it's unclear if it was related. It says possibly related. In J&J, they did see some urticaria, thromboembolic events, and tinnitus. And as everyone is aware, the US FDA halted J&J for about a week, and they studied these thromboembolic events. They found that there were a few cases, mostly in young people, more so in young women. However, the frequency was way less than what is seen with actual COVID-19 infection or birth control pills or smoking. So overall, very, very low case of thromboembolic events, and the J&J vaccine has been released and is being administered again. The only precaution is that we monitor patients after for any side effects. In Covishield, there were 3 cases of transverse myelitis reported, and this trial was halted for a little while in between. 2 were considered unrelated. 1 was thought to be idiopathic, so none related to the vaccine. Covaxin, again, their phase 3 trial data is not available. I could not really analyze it, but they claim that their vaccine was well tolerated in both dose groups with no serious adverse events. What are the advantages and disadvantages of each vaccine type? So mRNA advantages, they seem to provide robust protection, 94-95% protection, unheard of in any other vaccine so far. I mean, our flu shot that we've been taking for years has about 50% protection. So that tells you how good these vaccines are. They're easy and quick to design. Once you have your mRNA vaccine machinery in place, you just switch out the mRNA sequence and you can make a mRNA vaccine for any virus or microorganism for that matter. They are less prone to contamination. So you have to keep in mind when we make a vaccine, we have to worry about how to clean it and purify it, right? These are subcellular electron microscope level structures. You can't really 3D print them. You have to use cellular machinery to make them. So when we make these vaccines, they're being made in the lab in cell lines. Now with mRNA, you're merely making mRNA, purifying it and breaking off all the proteins. And then you're introducing the mRNA into the host body, and the host body can make its own spike protein. So you're literally just giving them the blueprint to make it, the recipe. As opposed to inactivated vaccines where you're making the whole virion in a cell line, you have to worry about what else that cell line is making, because that cell line that lives in your petri dish in the lab needs to survive on its own, so it makes its own proteins. So this is akin to the difference between giving your friend the recipe to bake a cake as opposed to baking a cake for them in your home and giving them the cake. So if your friend had a lot of food allergies and you are baking a cake in your home for them, you would worry about, did I contaminate it with egg? Was that spoon clean? Was that pan used for milk yesterday? What did I contaminate it with? Did I clean my kitchen properly? As opposed to that, you can just give your friend the recipe to bake the cake, and their kitchen is probably well suited for their needs and their food allergies, and they can bake their own cake, and it's always going to be better suited for them. So that's why these mRNA vaccines are like giving your body the recipe to make its own spike protein as best suited to you, versus trying to introduce spike protein made outside in a foreign cell line, and then you worry about contamination. In the long run, mRNA vaccines are cheaper to manufacture once you have the machinery and the process set up. What are the disadvantages of mRNA vaccine? They are new vaccines. They need to be frozen because RNA is very, very fragile. So we need to have access to freezers, which can be a big thing in a country like India, where your rural health centers may not have a ultra-low freezer to keep these vaccines frozen. What about viral vector vaccines? Advantages? They seem to offer good protection, 65 to 70 to 80% protection. They do not need to be frozen because these viral vectors are robust. You can keep them in the fridge. What are the disadvantages? They are new again. So viral vectors are also a new technology. You are going to mount an immune response to the vector itself. So if you need booster doses, we are going to have to change out the vector, because otherwise pretty soon when you get a viral vector vaccine, if you already have mounted a response to that particular adenovirus, then your body won't even get a chance to see the spike protein inside. It will just destroy the adenovirus because it thinks that that vector is the threat. And we have to make sure we always pick a safe vector that will be non-replicative, non-infective, and will still work to deliver the message to our cells. What about inactivated vaccines? Advantages: old technology, well established. We know this works, right? And there is no need to freeze it. They're stable in the fridge. Disadvantages: old technology. So it's like using a typewriter in the days of an iPad. There are disadvantages to using old technology. It's older, it's not up to date, it's not as effective. Another disadvantage: they require adjuvants, like we discussed. When you use an inactivated virus, you have to tag it with an adjuvant to make sure that the body recognizes that this is a threat and mounts a response. Otherwise, the immune system may just disregard it as trash and never mount a response. And the immune response may not be as robust because of these reasons. What about in pregnancy? So in general, women of childbearing age were not allowed to enter the trials if they were pregnant or planning on becoming pregnant. And that's because as a society, we are incredibly protective of our children, of our young ones. So we always try out new things on non-pregnant healthy adults first before we will give them to our children, or we will allow pregnant women to get them because it may affect our future children. However, in spite of everything, several pregnancies did happen in the trials because, this is life. Unplanned pregnancies happen even when one is in a trial. In the Pfizer trial, 23 pregnancies were reported after the women had already gotten the doses. 12 were in the vaccine arm and 11 in the placebo arm. There was 1 spontaneous abortion and 1 retained products of conception, both in the placebo arm. There were no adverse effects on the pregnancy from the vaccine in the vaccine arm. And as far as the data has been released, the rest of the pregnancies are progressing fine. In the Moderna trial, 13 pregnancies were reported, 6 in the vaccine arm, 7 in the placebo arm. 1 spontaneous abortion and 1 elective abortion, both in the placebo group. No adverse events in the vaccine group. In the J&J trial, 8 pregnancies were reported, 4 vaccine and 4 placebo. 1 spontaneous abortion and 1 elective abortion in the placebo group. 1 spontaneous abortion in vaccine group. 1 incomplete and 2 elective abortions in placebo group. 1 ectopic pregnancy in vaccine group. So again, no adverse effect from the vaccine on any of these pregnancies. The data is still not available for Covishield and Covaxin on exactly how many pregnancies were reported. After the trials, millions of women worldwide have chosen to get the COVID-19 vaccine during pregnancy, and whereas the vaccine adverse event reporting system in the US has not received any signals that this might be harmful. In fact, we are now finding that babies born to these moms that got COVID-19 vaccine in pregnancy are being born with ready-made COVID-19 IgG. So the babies are going to be protected for the first few months of their life, which is excellent. So in the US, the recommendation is that we encourage all pregnant women to receive COVID-19 vaccine during pregnancy. In breastfeeding, the Academy of Breastfeeding Medicine does not recommend cessation of breastfeeding for individuals who are vaccinated against COVID-19. And that is because there is little biological plausibility that the vaccine will cause any harm. And in fact, breastfeeding moms, once they mount a response, the antibodies to SARS-CoV-2 will be excreted in their milk and will provide some passive protection to their breastfeeding child. So again, the recommendation is to encourage all women who are breastfeeding to get vaccinated against COVID-19. So this data is to show you that these COVID-19 vaccines are highly effective, very, very safe, and this is going to be one of the big steps in overcoming this pandemic. So the way to get anything done is to get started. And this is one of the big steps that we need to start. So encourage everybody you know to get the COVID-19 vaccine when it is their turn. This is going to be one of the main tools in our arsenal, not the only one, but one of the main ones in trying to overcome this threat that essentially may wipe out our race if we are not careful. These are my references, and I'm happy to answer any questions. You can contact me. Thank you.